Hello and welcome back to the second IAS 2021 Prime session. I'm Eckhard van Hirschhausen, your Master of Ceremonies. We kicked off the Prime session this morning with a great talk by Professor Deborah Williamson joining us from Australia on STIs in the genomic era. If you missed it, you can watch all sessions again through the calendar. We now turn to HIV cure. 40 years since AIDS was first reported, we've had major advances. The Berlin and London patients who've been considered cured of HIV offered seminal breakthroughs for HIV science. There's promising research being carried out towards an HIV cure, but we have yet to discover a scalable cure. The journey is still very much ongoing. In this plenary, we will hear from Dr. Dana Duerk, Chief of the Human Immunology Section, Vaccine Research Center at the National Institute of Health in the United States on HIV reservoirs, lessons for cures. We then look forward to a live Q&A with Dr. Duerk, moderated by Professor Paula Cannon of the University of Southern California, United States. And, as in the first section, you can submit your questions through the Q&A chat box. Please do so. And now, take it away, Dr. Duerk.
Hello, thank you for coming to my talk, and thanks to the organisers for inviting me to present. I'm going to talk to you today about where we stand in terms of cures for people living with HIV. I have no conflicts of interest. Now, there are rare examples of HIV cure, what we might term eradication or remission. For example, in 2009, Timothy Brown received a hematopoietic stem cell transplantation from a Delta CCR5 donor and remained virus-free until he sadly passed away very recently. 10 years later, in 2019, Adam Castellejo underwent the same procedure, and there are examples of post-treatment control and profound elite control which might be thought of as remission. Now, even though these are successes, they will not likely translate into scalable approaches for most people. However, they are very important proofs of concept that an HIV cure is feasible. Let's begin by asking a few questions about the reservoir. But most importantly, which is the HIV reservoir that matters, that will cause damage when antiretroviral therapy stops? The question, where is the reservoir, can be answered relatively straightforwardly. You can see, for example, this beautiful paper from Jake Estes and Tim Shacker shows that 99% of infected cells in someone on ART are, as you would expect, in the lymphatic tissues, most notably the gut, and these cells are the source of recrudescent virus on therapy interruption. What is the HIV reservoir? The majority of integrated proviruses are defective and therefore not replication competent. Shu has recently shown that some competent viruses are integrated into gene deserts, areas of chromatin that are completely shut down. Other replication-competent viruses may be found in CD4 T cells that are in such a state of deep quiescence that they are unrousable. So these three likely do not contribute to the reservoir that matters. What does matter is this replication-competent virus that is found in a transcriptionally permissive site in a quiescent CD4 T cell that may become activated. Many investigators have asked, is there a phenotypic or transcriptomic profile of a quiescent cell containing replication-competent HIV that we can identify and target? And indeed, CD4 T cells that are enriched in HIV may be found among proliferating and activated cells, and T follicular helper cells, cells expressing exhaustion markers. Less mature cells are enriched for HIV, but also expanded HIV clones are found in more mature cells. But I think it's unlikely that we will find a marker or even a combination of markers of a quiescent infected cell other than the proviral genome itself. And it's often been said that looking for that cell is like looking for a needle in a haystack. But I think it's more difficult than that, because it's like looking for a needle in a haystack that is made of needles. And how big is this reservoir? We understand that fewer than 1% of published virus sequences are classified as intact using various assays such as near full-length PCR. They should have no deletions, no frame shifts, inversions, or hypermutations. The problem is that a virus may seem perfect according to the assay you choose to interrogate it. It may make transcripts that you can detect by RT-PCR. It may make proteins that you can detect by ELISA. It may even be induced to make virions that are infectious in cell culture. But if that virus contains an easily missed functional error in LTR or VPR, for example, then the virus is not replication competent in vivo and does not contribute to the reservoir that matters. So all that I feel comfortable in saying is that only a tiny and difficult to quantify fraction of HIV genomes are able to produce replication competent variants in people on ART. But you know, I'm not sure how important it is to know how big the reservoir is. Here is an example from Tim Henrich describing an individual who began ART on the day of exposure, who had no detectable reservoir that was thought to be less than 100 replication-competent virions. Yet 8 months after therapy interruption, a single virus emerged. So what is important is not so much the size of the reservoir, but knowing how to remove it and stop it from growing. And as no biomarker accurately predicts post-treatment control or time to rebound, treatment interruption should be the clinical endpoint of cure approaches. Which brings me to clinical strategies for an HIV cure. Now, we can target the infected cell, which contains the virus itself, or we could enhance immunity to kill that infected cell. A single approach may affect multiple of these pathways, and I think it's important to stress at this point that combination therapy is almost certainly going to be necessary. So let's first consider very early ART. It's clearly not curative. I just showed you this example from Tim Henrich, and here is another study of more individuals. We're clearly up against a wall here. If we were to consider ART any earlier, we would be in the realm of pre-exposure prophylaxis. I'd like to state that very early ART can be curative in non-human primates, but as I've just said, not in people. And so from now on, I'm going to predominantly focus on studies done in humans. Latency reversal, or shock and kill, is an approach whereby HIV transcription is reactivated with a latency reversing agent, such that virus is produced as a result, and then the immune system, or the virus itself, will kill the cell, and it's done under the cover of ART to stop new infections. Multiple LRAs have been tested in vitro, and in vivo they show increased HIV transcription, however no decline in HIV reservoir size. Activation appears to be inefficient, tissue-specific, cell-type specific, and specific to the transcriptional state of the virus. More potent, less toxic, LRAs such as SMAC mimetics show no change in reservoir size or delay in rebound. It seems that there is no kill of the infected cells, which are relatively resistant to death, and the virus, of course, has escaped the immune system. So the question is this a problem with these LRAs, or is it a problem with the approach in general? Have we reached a biological limit with non-specific LRAs? And perhaps we can add a degree of specificity to this approach. Which brings me to immune checkpoint blockade. Immune checkpoint markers are a variety of receptors on many different immune cells that act to dampen the immune response. Immune checkpoint blockade non-specifically enhances immunity, but within that immunity there is of course HIV specificity encoded in HIV-specific T cells and B cells. PD-1 and CTLA-4 are expressed on exhausted T cells in HIV infection, and they have been shown to suppress this immune response. Immune checkpoint blockade in vitro and in vivo also reverses HIV latency. And HIV is enriched in PD-1 positive cells in blood and lymph nodes from people on ART. So immune checkpoint blockade acts both to reverse latency and enhance immunity. However, results from clinical studies to date are variable and generally not encouraging, although I would say it is still quite early days. Here's an example of a phase 1 clinical trial of anti-PD-1 alone or with anti-CTLA-4 in people living with HIV on ART who had cancer. Now, HIV transcription increased in the recipients of anti-PD-1 and CTLA-4, and what you can see here is that there was a significant decrease in virus outgrowth in 2 people. So it's not as encouraging as we would have hoped, but remember, it's a start. We can try to enhance immunity specifically using vaccines, and in fact, many therapeutic vaccine trials have been completed over the past 20 years using many different vaccine modalities. They have been safe, generally immunogenic, but of no benefit in most studies and only modest benefit in a few studies. We understand some of the reasons why therapeutic vaccines may not work. There are pre-existing immunodominant responses. Virus epitope escape has occurred. There is lack of potency of the vaccines, and this is in a setting of overall inflammation and immunosuppression, as well as there being immune-privileged tissue sanctions. So there are many barriers to therapeutic vaccine efficacy that we understand, and indeed some of these may be overcome, for example, using immune checkpoint blockade. I'm showing you here the result of a trial that was presented at recent CROI, in which 45 individuals who began ART in acute infection were recruited to receive a vaccine consisting of DNA and MVA or placebo, were then rolled over to receive chimp adenovirus MVA vaccine or placebo, which was followed by a therapy interruption, and then ART was restarted. Now you can see in these results here that in the individuals who did not have protective MHCs, that there was a modest effect in that therapeutic vaccination induced a slightly lower virus set point. Which brings me to broadly neutralizing antibodies specific for HIV, many of which have been discovered over the past few years. I'm showing you at the bottom the names of these antibodies, and at the top I'm showing you the breadth of envelopes that are neutralized, a panel of 208 HIV-env pseudoviruses. The y-axis shows you the potency, and as you go down, the potency increases. So CD4 binding site antibodies are broad and potent. Those specific for glycans are less broad, but can be much more potent. The EMPA targeting antibody 10E8, very broad, not so potent, it met with some manufacturing difficulties. And when we combine specificities with combinations of antibodies or trispecific antibodies, they can be very broad and very potent. And this study in which 2 potent BNAbs were administered to chronically suppressed HIV-infected people showed that those with BNAb-sensitive virus remained suppressed for 15 to 30 weeks during a therapy interruption. If we consider the breadth of coverage of combinations of antibodies or trispecific antibodies, and we put it in the perspective of the recent antibody-mediated protection, or AMP trial, where at an IC80 of 1 microgram per mil, VRC01 only gave 29% coverage. If we look at the table on the right, then the percent viruses neutralized for any of these antibodies on their own is 49%, 63%, and 83%. The theoretical combination where only 1 antibody needs to be active against an envelope is 94%, if 2 active, 74%, and if 3 need to be active, 27%. The coverage for the trispecific antibody is 81%, considerably greater than that for VRC01. And clinical trials of these combinations are currently being performed by Boris Yule. 1 study has been completed, and another study consisting of 6 monthly injections in ART-suppressed people with a therapy interruption after the first dose of antibody is enrolled, and I'm really looking forward to seeing the results of that study. Now, an exciting way to deliver these BNAbs is to use the genes that encode them. One could perform ex vivo modification of B cells, or more simply, one could deliver the genes using viral vectors such as adeno-associated virus, AAV. The advantage is that transduction occurs in vivo after IM administration. The transgene persists in the nucleus and is stable in post-mitotic cells. And this experiment using AAV to deliver monoclonals performed in non-human primates showed sustained suppression of viremia after AAV delivery of BNAbs. Here are the results of a study presented by Joe Casaza at the recent CROI, where AAV vectors were used to deliver VRC07 to 8 HIV-infected people on ART. You can see the results in the graphs on the right. 5 of these 8 volunteers produced functional VRC07, which started 4 to 6 weeks after AAV administration. 4 had VRC07 concentrations at or near maximal for 1 to 1.5 years, and this is remarkable. This is the first trial to show the induction of a difficult-to-elicit broadly neutralizing antibody in humans by a therapeutic intervention. I mentioned at the beginning that I thought combination approaches are very much likely to be in the future of HIV cures, and there are a number of approaches combining LRAs with various immune interventions. And I'd like to draw your attention to this one from UCSF, sponsored by AMFAR, where the LRA is a TLR9 agonist, which also acts as an adjuvant for a vaccine, and that's given in combination with 2 bNAbs. That study is enrolled, and at the moment it's in the therapy interruption phase. Many other trials combining bNAbs with immunomodulatory agents are underway. However, significant effects on the HIV reservoir remain to be shown. Now, I said at the beginning of my talk, and many people say the same, that the examples of Timothy Brown and Adam Castellejo are proof of concept of an HIV cure. However, that's not quite right, because more specifically, they are proof of concept of an HIV cure by gene therapy. That is, the targeting of the host genome to disable expression of particular genes. One could disrupt the HIV proviral genome itself, or one could remove something that HIV needs, such as CCR5. And the tools available to do this include CRISPR-Cas, nucleases, shRNAs, and so on, and one can manipulate the cells ex vivo or deliver the agent in vivo. And I'd like to show you some data from a remarkable study recently posted to bioRxives by Rafiq Sekali and his colleagues, in which 15 people with HIV, fully suppressed on ART, underwent leukapheresis, from which CD4 T cells were enriched. Some of them obviously expressed CCR5. These cells were then treated ex vivo with a zinc finger nuclease that physically cuts the CCR5 gene. And what you get is a product consisting of CD4 T cells, some of which cannot express CCR5. These cells were then reinfused, and a therapy interruption was performed 6 weeks later. Importantly, this immunotherapy is minimally invasive with no severe adverse events. It's much more accessible than hematopoietic stem cell transplantation, and there is no need for compatible donors and no risk of GVHD. And the results show that 5 individuals partially controlled virus after treatment interruption, so we may call them post-treatment controllers. They had decreased virus reservoir size in terms of integrated total and intact provirus, and they showed persistence of CCR5-edited CD4 T cells. Interestingly, one individual controlled virus load for 5 years after infusion, and it's thought this may be due to a small replication-competent reservoir, persistence of CCR5-modified cells, and increased HIV-specific T cell immunity. I find these results very encouraging. So where do we stand, and where are we going in terms of an HIV cure? I think I've made it clear that it's still very early days. We have to understand there is a whole spectrum of how people living with HIV fare on ART. Some may be well with no problems of adherence or access. Others may not be doing so well for many various reasons. And within that spectrum, we must be able to offer a range of scalable and safe options. From a true cure with no virus, to various remission strategies, to the understanding that some individuals want to be left alone and are perfectly fine and would like to remain on their current regimen. Understanding the biology of persistence of the quiescent HIV reservoir is critical to discover targets. We have to test multiple approaches in non-human primates and humans, and by multiple, I mean combinations and lots of them. We must also not be afraid to call it quits for an approach which is going nowhere. This will likely be a long, bumpy, and iterative process. The first cure will not be able to compete with ART for most people. But I have no doubt that we will get there, because that's what we do as scientists, as doctors, as everyone associated with this effort. We make progress. It's our job. I'd like to thank you all for listening. I'd like to thank everyone in my lab, my collaborators, and especially Sharon, Steve, Rafiq, and Mirka for help in putting this presentation together. Thank you.
Thank you, Danny. That was a very thoughtful and thought-provoking talk. We now have some time for questions, so I'd like to encourage the audience to type any questions you have into the Q&A button. But while we're waiting for those questions, meanwhile, to get us started. So Danny, I was really struck when you showed the data from Jake Estes and Tim Schecker about where the reservoir is both on and off ART in their NHP study. And it's like, as you said, 99% in lymphoid tissue, especially in the gut. And it made me, I was really struck by how little of it there appeared to be in the brain. And so I wondered what your thoughts were about a reservoir, or the role of a reservoir that might persist in the CNS, and whether where the reservoir is even matters for some of the cure strategies you outlined.
Thank you, Paula. Thank you for your kind comments. I think where the reservoir is can matter if it's in a privileged site. I think one thing we understand about immunology now is that there are fewer privileged sites than we once thought. The brain was thought to be locked out in terms of the immune system, but we know that the immune system comes in and out of that. Now, the question you're asking specifically is really, what about monocytes, and more specifically tissue macrophages? Are they a reservoir for the virus? My feeling about this, which I've made very clear on many occasions, is that the reservoir that matters of HIV is in quiescent CD4 T cells. I do not think that macrophages or myeloid cells present an important reservoir that matters for HIV recrudescence of ART. However, I remain open-minded, and if I were to see evidence from people that really convinced me, then I would accept that. Now, if we find that macrophages are a significant factor in the reservoir that matters, and those are in the brain, and the brain is difficult to access for cure modalities, then the site of that reservoir becomes absolutely critical.
So you spent quite a bit of time at the beginning of your presentation talking about the reservoir size. And so contrary to everything I think I've ever been told before, apparently size doesn't matter. And I think you were sort of saying that it's really not a predictor of either control or even time to rebound. Is that a correct assessment of what you're saying?
That's correct. Generally speaking, it's not an accurate predictor. We don't have any accurate predictors of time to rebound or control. You know, I spent so much of my career measuring the size of the reservoir, thinking of a new assay, as do many of my colleagues, encouraged by people, you know, we've got to find the gold standard measure of the size of the reservoir. And then there's that paper that I showed from Tim Henrich. And there are other papers, the Boston patients who recrudesced. It's always one virus that comes back. And then I was thinking about it. I was trying to draw my slides, because I always used to show a slide of the size of the reservoir and how big it is and everything. And I was drawing it and trying to put numbers to it. And then I just thought, but if one virus matters, then why does the size of the reservoir matter? And I've come to realize that it really doesn't matter. And I don't think we should be spending so much time trying to accurately measure the size of the reservoir. And we should spend more time, as I said, trying to understand how to get rid of that reservoir. Because if all you need is one virus and you can't find that virus, then you can measure as much as you like, and it's not going to change your approach to a cure.
So then you also touched on some of the latency reactivation strategies, the LRAs, and does our approach to those need to be rethought? Do you think LRAs will ever be able to, get rid of the reservoir that matters, or are they always just going to be some sort of debulking approach that might be nice to have in combination with some other immune therapy, but really it's not going to get us where we need to be alone?
That's a great question, Paula. But when you say, will LRAs ever be used alone? I don't think any approach will ever be used alone. I think we're going to be looking at combinations. Even for the fact that some people will get side effects from one and not from another, and it'll be different in all people, I think we're going to have to look at combinations. So let's look at LRAs specifically. I think we have to accept that they have been a great disappointment. The majority of studies have shown an increase in the amount of virus in individuals who've been treated, not a decrease. Now, obviously, you want to try and get expression of the virus, but you also want the result of that to be a decrease in the size of the reservoir, and that hasn't happened. So I asked the question, is it a problem with the approach overall, or is it a problem just with the LRAs we've been using? I think what we have to realize is that there are some cells which are very, very difficult to wake up, infected cells, T cells, which are very difficult to wake up. And if we are looking for even stronger and more potent LRAs, then we're going to run into the trouble of toxicity.
Right.
Now, SMAC mimetics are very potent with less toxicity, and they've shown no benefit either. So does it mean that I think we should quit completely? No, because one thing I've learned is certainly from in vitro experiments, you can never predict what is going to happen in vivo. And I am a real champion of doing as many phase 1 studies of combinations of different things as possible, because that's the only way that we're going to learn. But I think we have to stop looking at LRAs on their own as a cure approach and start thinking more intelligently, as people are doing, as I mentioned in my talk, about how we mix and match LRAs with a whole host of other interventions. So I think that's the future, if anything, for LRAs.
So maybe as a step further than that, of course, you touched on it very briefly, but people are thinking about using genetic tools such as CRISPR-Cas9 to actually target an integrated provirus and disrupt it. And there the idea would be that regardless of whether this was a replication competent or wakeable virus, you could still kind of do damage. But of course, you also touched on the fact that the big challenge there is how do you deliver such reagents such as CRISPR to cells that contain even any HIV, let alone a reactivating HIV. And I think you talked about looking for a needle in a haystack full of needles. Ouch. So I'm guessing you're probably not a fan of that approach, at least with the current technology as well.
Which of the approaches?
Well, the idea of packaging something like a CRISPR-Cas9 that targeted an integrated provirus into a vector and injecting that into somebody and hoping
that. I mean, I mentioned CCL5 knocking that out, obviously, but I really do have an extreme fondness for these genetic approaches, and if the problem is delivery, I'm sure we'll overcome it. So this is something that I didn't say in my talk, but I think I should say now is I think we need to understand, we have to be honest with ourselves. We're a long way off from a cure. We really are a long way off. But if I think 10 or 20 years' time into the future, I can really see delivering an enzyme to specifically genetically modify the provirus. I can see that in the future. As I said, I have a great fondness for those approaches because they're highly specific. And if we can figure out the delivery of those mechanisms, I think we'll have a great cure approach. And I see that coming. It might take a long time, but I think it'll happen.
Yeah, and I think the good news is that the idea of in vivo delivery of genetic tools is an area of intense research for all sorts of other diseases, genetic diseases in particular. So to some extent, the HIV cure field is likely going to be able to take advantage of the advances that are coming from other diseases as well and adapt them to target.
I think you're so right. I mean, we learn from cancer, from immune checkpoint blockade, for example. The other fields learn from HIV. The recent the ongoing COVID pandemic, and the incredible thing that's been done with vaccines against SARS-CoV-2. A lot of that came from HIV and what we could give to the field. So this exchange of ideas between different fields of biomedical science will help. And it'll help in such a way that we will see approaches to cure improving or amassing, not in a linear fashion, but I think in an exponential fashion.
Yeah, and I think I would just agree with you there and say, I think, I'm always struck in our field by the combination of the innovation of the scientists, the support and the push, from the advocates and the people with HIV, and also to some extent the bravery of the funding agencies. I mean, it's in many ways it can be a perfect storm, and while the challenge of an HIV cure remains daunting, I think, the as you say, the benefits that we have for other diseases is real.
I have no doubt we will get there. I really, as I said in the end of my talk, we'll get there because we make progress, because it's our job. It's what we do. It'll take us some time, but we will get there.
I want to cure HIV before we land on Mars. So there you go. Okay, so maybe I'll switch to talking about broadly neutralizing antibodies or BNAbs. You showed some very exciting data there from a couple of different studies that BNAbs, when delivered as either an injected recombinant protein, or even more impressively, when expressed from AAV vectors that were, I think, injected into the muscle, can actually suppress HIV long-term, as long as you have the right BNAb or combination of BNAbs for the virus within an individual. So for the AAV BNAb data that you showed, Joe Casave's data, yeah? Is an ATI planned in that trial to see if the BNAbs can actually suppress HIV?
I don't know. I should have asked Joe before I gave my talk, but I would be surprised if an ATI were not planned, because that's the whole point of doing it. It was a remarkable study, and I think that bodes well for the future.
I think so too. And then when you showed Joe's data, I think was it 5 out of 8 of the individuals maintained high levels of the antibodies over 1 to 1.5 years. But I think for the 3 individuals that didn't do that, it was because they had developed what we call anti-drug antibodies, basically antibodies against the antibodies, which is kind of annoying because, of course, they're sort of foreign to the individual. Do you have any insights into how much of a problem you think such anti-drug antibodies might be, or how that could be addressed?
It's always a problem with any drug, especially biologicals such as these. One way of addressing them is by increasing the dose. So you have a level of your therapeutic antibody, you may have a level of your anti-drug antibody, but if the subtraction one minus the other is enough to be therapeutic, then you're okay. It's also possible to engineer antibodies in such a way to make them less visible to the immune system. And there are lots of clever ways of playing with immunity to make that less of a problem. So I think that needs to be, it is being addressed in the future. But I think, of all the problems that one could have, that's a problem that's definitely dealable with.
Yes, and again, the gene therapy field would agree with you. I think you can tolerize by having some expression from the liver, for example. That seems to be one strategy that's been used in other approaches.
Yes, that's absolutely correct, yes.
Okay, so while we touched on immunology, and you're an immunologist and I'm not, I'm going to ask you sort of a more naive question. So when I see these effects of broadly neutralizing antibodies, suppressing the virus, and I think about the name broadly neutralizing, it says to me it's an antibody and it's going to stick onto the virus and neutralize it and just, stop it being able to infect a cell. But of course, antibodies do more than that, and they interact with other parts of the immune system. So perhaps you could talk a little bit about, what an antibody actually does beyond just sticking to a virus.
Right. I mean, so antibodies stick onto the top of the envelope and they block the interaction with the receptor. Right. That's one thing. But antibodies have another part to them, their tail end, which is the FC, which binds various receptors in the immune system. And that results to an enormous amount of downstream effects that are mediated by many things, among them cells, such as macrophages, B cells even, and also NK cells. So an antibody binding to a virus, or an antibody binding to a virus expressed on an infected cell, can result in an enormous, varied immune response that probably plays a role in the effect of the broadly neutralizing antibodies. And again, playing around with the FC and its effects on other immune cells, there's a sort of whole world of stuff that you can do to these antibodies as a therapeutic agent.
Mm-hmm. Another approach, you had it in a bullet point, is the idea that also these antibodies now, several groups, including it's something my own group works on, are figuring out ways to take these preformed BNAbs and actually express them from the authentic immunoglobulin locus in B cells. Yeah. And what's kind of cool about that is, first of all, it's very, very difficult to do that because the immunoglobulin locus is huge and complex. But once you've done it, you then get to not just produce an antibody, but you can tap into all the other benefits of natural antibody production, including, being boostable, both by the presence of HIV as well as, a sort of vaccination. So I wondered if what you thought about that, about actually using B cells themselves to express the antibody rather than the muscle.
I think it's great. We're not going to end up with one approach. We're going to end up with a ton of different approaches. The thing I like about sticking AAV into a muscle is it's an injection. Mm-hmm. So it's easily scalable. Right. But you have problems with anti-drug antibodies. Modifying the genes of a B cell, you have to do that, well, certainly initially, you're going to have to do that ex vivo. Take some cells out, modify them, put them back. So that's not going to be as scalable. But in the future, maybe it's going to be pretty scalable. And I think that will be one of the things in the armamentarium that we'll have for cure approaches. Different approaches, different combinations of approaches for different people in different settings all over the world.
Mm-hmm. That'll keep a lot of us scientists busy then, won't it?
Oh, yes.
Okay, so we've spent quite a bit of time talking about antibodies. So I want to switch now and talk about some of the sort of the approaches you talked about around the CCR5 editing. You, of course, talked of the examples of Timothy and Adam, who underwent bone marrow transplantation to receive HIV-resistant, naturally CCR5 negative cells, and the idea that instead that can be recapitulated in an individual's own hematopoietic stem cells or T cells. So the bioarchive preprint from Rafiq Sekali's group that you pointed to, that's really exciting, I think, and especially the fact that suppressing HIV correlated with long-term persistence of CCR5 edited T cells. Can you say anything about the hypothesized mechanism for the long-term persistence of those CCR5 edited T cells?
Yeah, this is really important, Paula, because it seems that the cells which were persisted obviously were CCR5 edited. That's great. But there's another thing, is that they were part of this subset of CD4 T cells that are known as stem cell memory cells. And they are a CD4 T cell which is at the kind of young stage of the maturation process of the T cell. And what they have is this great capacity for longevity and for proliferation. Now, longevity, they last a long time, which is great. Proliferation means that they can supply the rest of the immune system. Why it's really important is we know now, we have an idea which type of cell it's important to get the gene editing into. So that gives us a lot of information for the future and how this goes on. Not only are they stem cell-like and they proliferate, they don't die, but of course, because they've got the CCR5, they're not infectable if they were to make an immune response, if they were to be HIV specific. So there's a whole host of attributes that these cells have that make them the ideal candidate for the cell to be edited, to carry that edit.
So you'd see the idea of rather than just editing bulk T cells from an individual, actually isolating or even ex vivo expanding I think they're called TSVMs. I think that's the
correct.
Yeah, the TSVMs ex vivo, or maybe in the future we could even have an allogeneic, sort of an off-the-shelf T cell that
Or you could give certain cytokines such as IL-7, which we know preferentially makes those type of cells expand. So it really broadens our horizons in terms of how we can manipulate the immune system to ensure that the gene-edited cells have an advantage and form the majority of the cells in the individual.
Yeah, no, that's kind of exciting. And I think just the advances in, again, this is HIV and HIV cure research really coming to the fore and teaching us about, 1,000 different things that all these different types of T cells do, and how we can sort of play them off against each other, so to speak, and have a combination, even just a combination cell therapy that has these different elements.
Yeah.
Right. So maybe the last question I'll ask you, we'll come back to what we talked about at the beginning, which is about kind of the size of the reservoir and everything. And I'm coming back to Timothy and Adam's cure, because in addition to having the CCR5 edited cells, they also, of course, underwent bone marrow transplantation for an underlying cancer. So they went through incredible, ablation of, chemotherapies, radiotherapies to get rid of not just their cancer, but that also got rid of presumably, 99.9% maybe of their HIV reservoir. So do you think it will be possible that just doing the CCR5 editing part will be able to recreate their cures in the absence of such reduction of the reservoir?
I think that we need to see if that's possible. Because as you said, what Tim and Adam had is a real insult to the system. It's really difficult. If we can get away with just gene editing cells, and then the virus may kill off some of the other ones instead of giving chemotherapy, radiotherapy, what have you, or maybe they never come out of quiescence. I think we have to do that. Maybe we'll have to have some kind of lymphodepletion at the same time as well, and the extent of that, maybe that needs to be decided on an individual basis. But Paula, honestly, that is the sort of knowledge that is going to come out of lining up one phase 1 study after another until we get it right for each person.
Absolutely. Well, I think on that note, we'll end. Thank you so much, Danny, for chatting with me. Danny's asked me to let you guys know that if you have additional questions, please just reach out to him personally. His inbox will now get flooded. And now we're moving on to a session where we're going to announce the winner of the IIS Abivax Research Cure Academy Fellowship Prize. Thanks you all for your attention.
Thank you.